So something very fundamental about your brain cells is that your brain cells have preferences. All right, so that's a drawing of your brain. It's essentially formatted as if I'm looking this way, so this half is like the front. So the areas of your brain that process vision are kind of localized somewhere over here. I'm gonna go for NT, which is the area of your brain that processes motion. So let's say I've opened up your brain and I have stuck a very thin needle called an electrode into that area MT. The needle is so thin that if this is your brain, if I magnify how it looks like on the scale of your brain cells, if this is a brain cell, like the needle is right outside of your brain cell, okay? And so it can pick up all of the electrical activity of your brain cells. And so let's say while I have this electrode right by one of your brain cells in the area MT, you are currently watching a bunch of dots moving in certain directions, right? Like there's a bunch of dots moving upwards, downwards, sideways, or whatnot. And so if I kind of try to figure out how this neuron is behaving while you're watching different directions, that gives me this plot up here. And this plot is what we call a tuning curve. On the x-axis, you have how much your neuron is firing action potentials to its neighboring neuron. And on the x-axis is the direction of the motion of those dots. What you'll find is that when you were watching the dots moving upward, the neuron was usually firing the most. And then as the movement of the dots veered away from the upward direction, your neuron was firing less and less and less. And so that is what we call a preference for upward motion direction. And this is a property of neurons throughout your brain. Now let's say I push that needle a bit deeper into your brain and then I start recording from this neighboring neuron instead. And then if I plot its firing rate, what you'll find is that for neuron number 2, the tuning curve is slightly different. It can be the same, but it will be slightly different usually. And over here, if you extrapolate this neuron really likes motion when it's rightward. And that is how we figure out that your neurons in your brain have preferences. Now it's very neat and clean and in these sort of more sensory areas of your brain. But when you move into other areas, such as your frontal lobe and even temporal and your motor cortex over there, that's when the story gets a bit more complicated. For example, a neuron from that area over there might be sensitive to the shape or color or motion direction or even a position of an object all together at the same time. And so when you're recording from that neuron over there, it will kind of behave almost like randomly, and it'll be hard to really tell right off of just observing it that what does it really prefer.